My name is Frankie Crutcher, and I'm the plant pathologist at the Eastern Agricultural Research Station. Today I will be discussing the small grain disease Fusarium head blight, including a brief background of the disease, management options for producers, and summary of current research being performed in Montana. Fusarium head blight, also known as FHB or scab, is a disease of small grains caused primarily by the pathogen Fusarium graminearum. The effects of the disease can most often be measured by the production of the mycotoxin deoxynivalenol, also known as DON, vomitoxin, or sometimes VOM. For many years, Fusarium head blight was only an intermittent problem in our state, but with the increase in corn acres and no-till practices, outbreaks have become more severe. This is because corn is an alternative host for the pathogen, and growth of the fungus on corn residue produces large amounts of orange spore masses that can be splashed into the canopy by rain and overhead irrigation. Other structures produced on corn and grain stubble, called perithecia, pictured as small black dots on the residue, release spores which can be dispersed by wind many miles from the field. When scouting for disease in the field, Fusarium head blight is recognized by partially bleached heads for wheat and purple to red discolored florets in barley. In some wheat varieties, infected florets will appear red or pink in color. Frequently, orange or pink structures can be found at the base of the floret. Under conditions of very high humidity, perithecia can also be observed. Scabby seed is produced in infected heads and are usually smaller, misshapen, and may be white or pink in color. If you suspect you may have Fusarium head blight, you can send samples to the Eastern Ag Research Center or to the Scudder Diagnostic Lab located on the Montana State University campus. Additionally, there is a quick and dirty disease diagnostic method you can use at home. Simply put the suspected infected heads into a Ziploc bag with a slightly damp paper towel and wait 24 to 48 hours. If Fusarium is present, pink or white fungus will be observed growing out of the head. Management of Fusarium head blight requires an integrated approach as no one method can give complete control. Minimizing cereal and corn stubble, either by tillage or by increasing rotations with non-host crops, is one of the most important methods of control, as it minimizes the amount of pathogen in the field. Additionally, the ideal time for infection is at flowering, which is when the anthers are bright yellow for wheat and when the head emerges from the boot in barley. Therefore, avoiding irrigation at this time can minimize infection. Farmers should use resistant varieties when available and apply fungicides if weather conditions are right. All labeled fungicides, including Pusara, Carumba, Proline, and Miravis Ace, have all shown approximately 50% control of vomitoxin production and disease symptoms, so employing other methods of disease prevention is essential. Strobilurin fungicides, which are the FRAC 11 fungicides, should not be used after flag leaf, as this has been shown to increase vomitoxin levels. The best timing for fungicide application is at flowering in spring wheat and head emergence in barley. However, recent research performed through the U.S. Wheat and Barley Scab Initiative has shown that this window can be extended 4 to 6 days and still have good disease control. Fungicide application should be performed with nozzles that spray both back and forth so that all sides of the head can receive good chemical coverage. Although cleaning seed to remove scabby kernels can lower vomitoxin in the final harvest, we do not recommend that the fan speed on the combine be turned up in order to blow the scabby seed into the field. This can result in increased amounts of pathogen, both in the soil and on the residue, which can cause root and crown rots in many plant species. Resistant varieties are also an important part of integrated pest management of Fusarium head blight, and our program is actively working with small grain breeders from MSU, Drs. Jamie Sherman, Phil Bruckner, Mike Giroux, and Jason Cook, to screen varieties for resistance to the disease. In order to get the maximum amount of disease for screening, we have constructed disease nurseries at both Eastern and Southern research stations, the former in collaboration with Drs. Ken Kephart and Jason Cook. Because the environment plays such an important role for infection of the pathogen, and the infection window is different for each variety being tested, in these nurseries, the ideal conditions for disease are maintained continuously. Each year, our program produces Fusarium inoculum, which is grown on water-soaked and sterilized corn and then dried in paper bags in the greenhouse. Our program plants both small plot and hill plots each spring, and we spread the inoculum in the field approximately 3 to 4 weeks from heading. This allows time for the fungus on the corn to produce fungal structures used to infect our experiment. As Fusarium head blight is harder to replicate for barley, even under these conditions, we also apply fungal conidia using a backpack sprayer. We make this application near dusk, as this has been found by other researchers to be the optimum timing. To maintain the higher humidity required for both infection and vomitoxin accumulation, we have also constructed misting systems at each site to regularly apply water to the field. Regular water misting increases humidity, essential for producing disease symptoms. I would like to thank the EARC Plant Pathology Program staff and students for all their hard work this year. Both Fusarium head blight nurseries and the MSU breeding effort are financially supported by the U.S. Wheat and Barley Scab Initiative.